Hi, my name is Doreen and I'd like to lead you through an investigation in which we electrolyze copper chloride solution. For this investigation, we will need copper chloride crystal, water, a battery or cells, 2 carbon electrodes, and connecting wires.
Solid copper chloride is a green-colored ionic crystal. It consists of copper ions and chloride ions held in a rigid crystal lattice. When copper chloride is dissolved in water, a copper chloride solution results. The crystal lattice dissociates. It breaks up as water molecules dismantle it. The copper chloride solution consists of copper ions, Cu2+, and chloride ions, Cl-, with each ion being surrounded by water molecules. The positively charged copper ions give the solution its blue color. Positively charged ions such as these are called cations. The negatively charged chloride ions are colorless. Negatively charged ions such as these are called anions. These can be oxidized. This means that electrons can be taken away from them. In the process, the chloride ions change into chlorine molecules.
So how do we do this? We need to pass an electric current through the copper chloride solution. To do this, we will need an external power source, leads, and electrodes.
The copper chloride solution will serve as the electrolyte. If you already have 2 carbon electrodes, you could use those. Otherwise, you can get some by dismantling 2 cells. If you don't know how to do this, watch the battery dismantling movie. Pour some copper chloride solution into a beaker. Put your 2 carbon electrodes into the copper chloride solution. Use the leads to connect one electrode to the positive terminal and the other to the negative terminal of the battery. What happens? After a while you will smell chlorine's characteristic sharp smell. Notice the bubbles of chlorine gas which form at the positively charged electrode. The chloride ions are being oxidized to chlorine molecules here. This electrode is called the anode because it is the electrode where oxidation occurs. Meanwhile, blue copper ions which had been dissolved in the copper chloride solution are reduced to reddish-brown copper metal atoms at the other electrode. We call this electrode the cathode because it's where reduction occurs.
Now that you've seen the basic structure of the electrolytic cell and learned the background theory, you need to plan an investigation you could do with the cell. Every experimental investigation must have an independent variable and a dependent variable. Let's start with the independent variable. We must have at least 2 treatments.
The independent variable is the characteristic by which the different treatments must be made to differ from one another from the start.
So we need at least 2 electrolytic cells in this investigation. These cells must differ from each other in one way, and that is the independent variable. How many different ways can you think of that we could make these cells different? Pause this movie until you've discussed this, and then fill in the relevant sections of the accompanying worksheet.
We could make one treatment hot and the other cold. Then our independent variable would be temperature. We could place a high potential difference across one and a low potential difference across the other. Then our independent variable would be potential difference. We could make one treatment's copper chloride solution concentrated and the other dilute. Then our independent variable would be electrolyte concentration. We could place our electrodes close together in one treatment and far apart in another treatment. Then our independent variable would be distance between electrodes. We could insert the electrodes almost all the way into the electrolyte in one treatment and only just in the electrolyte in the other. Then our independent variable would be electrode surface area in contact with the electrolyte. We could use a large volume of electrolyte in one treatment and a small volume of electrolyte in the other. Then our independent variable would be electrolyte volume.
What is the reason for treating our treatments differently? We would like to see how this affects another variable, the dependent variable. The dependent variable depends on the independent variable.
In an experimental investigation, we expect the treatments to become different after a while because of having treated them differently from the start.
What is the dependent variable in these electrolytic cells? Pause this movie and discuss this with your teacher and classmates.
The dependent variable in this investigation is rate of electrolysis. Rate of electrolysis means how much of the electrolytic reaction happens in a certain time. Now we need an indicator of this variable, something we can measure which will tell us about electrolysis rate. Pause this movie while you discuss at least 3 different ways we could measure rate of electrolysis in this particular cell. As a hint, think of what happens and what changes in the cell as the reaction proceeds. Chlorine gas is produced at the anode. If the rate of electrolysis is high, a lot of chlorine is produced in a certain time. If the rate of electrolysis is low, little chlorine is produced in a certain time. We could measure volume of chlorine produced in a certain time, for example in 5 minutes, as our indicator of rate of electrolysis. Copper is deposited on the cathode. We could measure the change in mass of the cathode in a certain time as our indicator of rate of electrolysis. As the blue copper ions are oxidized, the electrolyte solution becomes paler in color. We could measure the change in electrolyte color in a certain time as our indicator of rate of electrolysis.
An experimental investigation needs to be focused by a question.
We can give this question in the form, how does independent variable affect dependent variable? In this investigation, rate of electrolysis is our dependent variable. If we choose to make our treatments differ in temperature, then temperature is our independent variable. Then our focus question would be, how does temperature affect rate of electrolysis? Give the focus question for each of these investigations. Here are the answers. How does potential difference affect rate of electrolysis? How does electrolyte concentration affect rate of electrolysis? How does distance between electrodes affect rate of electrolysis? How does electrode surface area in contact with the electrolyte affect rate of electrolysis? How does electrolyte volume affect rate of electrolysis?
So far, we've discussed a number of different investigation possibilities. You can try any of them.
For this movie, we have chosen the focus question, how does potential difference affect rate of electrolysis? So we must have at least 2 treatments, and these must differ in the potential difference we apply across the electrodes. Actually, we will use 4 treatments rather than 2.
A hypothesis is a scientific guess. It is what your current understanding of the theory tells you the answer to the focus question should be. Pause this movie and come up with a hypothesis. Answer the focus question according to your understanding of electrolysis. Justify your hypothesis. Why do you think it's correct? For a test to be fair, the treatments must differ only in the independent variable. All other variables that might affect the dependent variable should be kept the same. we say that they are controlled.
In our investigation, our 4 treatments must differ in the potential difference we apply across the electrodes. That is, in our independent variable. How should the 4 treatments be the same?
Pause this movie and discuss this, and then fill in the relevant sections in the accompanying worksheet. We must ensure that all treatments have the same temperature, concentration, distance between electrodes, electrode surface area in contact with the electrolyte, and electrolyte volume. We should also use the same type of electrolyte in all cases, as well as the same material for the electrodes. We should also apply potential difference across each for the same period of time.
We are going to electrolyze each of our cells for 2.5 hours. We are going to use the change in mass of each cell's cathode during this time as our indicator of rate of electrolysis. This mass change happens because copper is deposited on the cathode. We need to measure the mass of each of the cathodes before the reaction starts so that we can compare this to the mass after 24 hours. We record our data clearly for each cathode. These are called the cathode's initial masses. Now we perform the investigation. Here we have our 4 treatments. Each has a different potential difference across it. This one has 1,5 volts across its electrodes, this one 3 volts, this one 4,5 volts, and this one 6 volts. We connect all our cells simultaneously and allow the cells to work for 2.5 hours. We then disconnect all the cells and allow the cathodes to dry overnight. Now we need to measure the mass of each cathode so that we can calculate the mass of copper deposited during the reaction. We record our measurements. These are called the final masses of the cathodes. We find the change in mass of each cathode by subtracting the initial mass from the final mass of each cathode. This tells us the mass of the copper which was deposited. We see that most copper is deposited on the electrode connected to the highest potential difference. The least copper is deposited on the electrode connected to the lowest potential difference.
Our conclusion is our answer to the focus question in light of results.
The focus question of this investigation was, how does potential difference affect rate of electrolysis?
Pause this movie and answer the focus question in terms of your results.
We have seen that a higher potential difference causes a higher rate of electrolysis. We could say, increasing potential difference increases rate of electrolysis.
How does a conclusion compare to your hypothesis? Can you explain the conclusion? A higher potential difference is associated with a greater delivery of electrical energy to the circuit.
In an electrolytic cell, electrical energy is converted into chemical energy. A higher rate of delivery of electrical energy to the cell results in a higher rate of chemical reaction.
Therefore, increasing potential difference increases the rate of electrolysis. Now that you've observed this investigation, why don't you get your own equipment and do your own investigation? You could either repeat the investigation watched or perform any of those discussed in the planning phase. If you don't have copper chloride solution, you could use blue window cleaner. Although the reaction won't be identical to what we've explained, there will be similarities.